Let's talk about information packaging. As you know, DNA encodes the entire information to build an organism, and all life on this planet uses DNA as a storage medium. Now, DNA is an incredibly long and thin molecule that has to fit into an impossibly small cellular compartment, the nucleus. And just to give you an impression of what we're dealing with here is, if the nucleus were the size of a golf ball, you would have to fit 10 kilometers of an incredibly thin thread into the nucleus, while at the same time being able to access the information that is encoded on the DNA in a meaningful manner. So this is a really formidable problem, and some of you in the room might be familiar with the challenges of storing information in linear form. And the challenges are that we have to protect it from physical damages and tangles. Your ABBA song just simply won't play anymore in a cassette tape that looks like the middle one. We need to accurately duplicate it during cell division. We need to find the required information to invoke gene expression programs, and we need to physically be able to access the stored information. In all multicellular organisms, I should say that malfunction of these processes results in cancers and many, many other diseases. Now, in all eukaryotic organisms, all higher organisms, this feat is accomplished by wrapping short segments of DNA around small protein particles to form nucleosomes, which are then further compacted. We now have a quite exquisite understanding of the structural detail of DNA organization in the form of nucleosomes, and you don't have to be a structural biologist to appreciate the fact that you need elaborate machinery to maintain the compaction and to regulate the access to the information in processes that are collectively termed as epigenetic regulation of gene expression. Now, it turns out that the histones, the nucleosomes, and the entire nucleosome management machinery is extremely conserved across all higher organisms. And this indicates that it was an essential component of us becoming eukaryotes. So this raised the question, who provided the nucleosome starter kit? Where does this all come from? And so we decided to look across the evolutionary tree, look for histones in organisms that arguably contributed to formation of the first eukaryotes. Those are archaea, those are small bacteria-like organisms, single-cell organisms, those are bacteria themselves, and also ancient DNA viruses that are happy purveyors of genetic information across domains of life. So let's talk about viruses first. We found that some giant viruses encode their own histones, and these histones form nucleosomes that are remarkably similar to the nucleosomes that we observe in higher eukaryotes. And they organize the DNA in the viral capsid, and you can observe the similarity when we superimpose it with a human nucleosome. We also looked in archaea. Archaea, the vast majority of archaeobacteria or archaea encode one single minimalist histone. And this histone can form nucleosome structures that are remarkably similar to eukaryotic nucleosomes, except for rather than forming defined particles, they can form slinky-like assemblies that of varying size. They just keep going, wrapping the DNA around them. Archaea inhabit every niche on this planet, and they live in often extreme environments. And we now suspect that histones have evolved in various archaea to adapt to these various conditions to help protect the DNA, which is the primary function for histones in these ancient organisms. Finally, we've also discovered that about 5% of all bacterial genomes known to date also encode histone-like bacteria. And it is these histones that are perhaps the weirdest ones, and they have the most variability in terms of how they organize DNA. We found one particular instance where the histone binds end on, and rather than wrapping the DNA around itself, it encases the DNA to form a nuclear histone filament that completely occludes the DNA from the solvent. Now, this really reverts the paradigm of what we currently have known, what we thought we knew about histones, in the sense that the DNA is straight, the histones wrap around them, and this flies in the face of histone logic, where normally the histones are wrapped around, the DNA is wrapped around the outside of the histone core. Now, we've only just begun to scratch the surface. There's an incredible variability of life on this planet, and we think there's a lot more to discover in terms of how these various and very often extreme organisms organize their genomes. And I think this will help us eventually to understand the principles, the primordial organization of larger genomes, and will help us to hopefully construct as a minimalist system that helps us to manipulate such genomes. Just to get back to the introduction, as you mentioned, constructing artificial organisms. I want to finish with one last thought that maybe has been alluded to but not explicitly said. Us scientists, we produce amazing science. We've heard a lot of examples from all the laureates here. But the second product that we produce is actually scientists. We train scientists, we train the next generation of scientists, and I think it's that latter part of our job that perhaps has the more lasting impact on society at large. Thank you very much.
Most of you probably have heard about this DNA methylation clock. So this clock is running in most of our cells, if not all of our cells. And the ticking of this clock correlates with gene expression changes and also correlates with the aging process. So well, that's just correlation. Whether the DNA methylation clock controls how long we may live is still debatable. So we ask this question: can we turn back the clock, DNA methylation clock? If we do that, can we slow down aging or even let us become young again? So 24 years ago, we happened to discover the first DNA demethylase. In the last session in this room this morning, we heard really interesting speeches about how we as humans sense the environment. Well, I've been studying how plants sense the environment, temperature changes, water availabilities, soil salinities. And during the process, we discovered whereby serendipity, the DNA demethylase. We've heard a lot of discussion about curiosity-driven science. Well, I think this is really another example of curiosity-driven science. And since we discovered it, we've been studying this whole process of DNA demethylation and how it's regulated, but we never thought it might be of any practical use until recently. So this protein ROS1 we discovered can recognize this methylated cytosine base and remove this base. And in the process, the DNA backbone is broken. And the basic season repair machinery comes in to fix this damage to the DNA and replaces the missing base with an unmethylated cytosine. So it's a complicated process. In the beginning, nobody wanted to believe this. But it turned out, this is the only way. In humans, 8 or 9 years later, the TET enzymes were discovered. So in humans, we don't have this enzyme that can recognize the 5-methylcytosine to directly remove it. The TET enzymes have to oxidize the methylcytosine, and then the basic synthesis repair takes care of the rest. So now, with the CRISPR-Cas technology, we can fuse the DNA methylation enzymes and DNA demethylation enzymes with the Cas, the inactive Cas proteins, and then guided by the guide RNAs to do targeted DNA methylation modulation or editing. So now we have not been, been there to inject into ourselves, but we injected just one shot of this lipid nanoparticles in containing these mRNAs for the fusion proteins and the guide RNAs into mice. You can see that after more than half a year after this injection, we look at these mice, and those that we injected with our DNA methylation modulators, they show clearly much slowed aging, as seen with alopecia and also cognition tests and also body temperature and many other biochemical and physiological tests. So this DNA, so it seems there is hope that modulating DNA methylation clock may be able to help us slow down aging, maybe in some day to reverse the aging process. In the meantime, we also tested these DNA methylation modulators in curing certain diseases, like in treating hyperlipidemia. So one shot of this DNA methylation modulator gives you sustained reduction in this target PCSK9 level in the blood and reduces the blood LDL cholesterol. So in theory, this can last for a whole life. One will not have to worry about the cholesterol problem anymore. So there have been a lot of studies in recent years on the aging process, and a lot of work has been done to intervene with the aging process, including gene therapies to correct the mutations, and mostly, I think, most importantly, to correct the epigenetic instability problem. So, and also there are G-cell therapies. There are lots of chemical drugs being tested, and also probiotics. But I want to draw your attention today onto the dietary intervention, which I call the food medicine. I really like this saying from Hippocrates, let your food be your medicine and your medicine be your food. I especially like the first part. I want our food to be our medicine. So a lot of studies show that healthy diet is really important for our healthy lifespan. And I think there are many problems in the modern diet in many parts of the world. I summarize here 3 major problems in modern diets. One is too much refined carbs, and secondly, there are too much unhealthy oil, unhealthy fats. And then thirdly, there are too little anti-inflammatory, antioxidants, and anti-aging phytonutrients in our food. So for the first problem, I think we need to consume more whole grain cereals. We need to make our cereals more nutritious. So I think we have created the most nutritious rice in the world. How did we do that? Well, the nutrients we really need, not, the starch in the grain, but it's on the surface of this rice grain, this layer called the single layer of cells called the aileron. It's rich in proteins, in minerals, in vitamins and dietary fibers. And by editing the gene, the ROS1 gene I just introduced to you, we can change this one single aileron layer to multiple layers. In this process, the nutrition that we need significantly increased. So to address the problem with refined carbs, we also created various low GI food, low GI rice, low GI potato, low GI wheat, low GI corn, really effective in blood sugar control and good for gut health. And the second problem with the unhealthy oil we have, so I think the most vegetable oil we have, like soybean oil, there's not enough oleic acid, omega-9. There's too much omega-6. The olive oil that recognized to be really healthy has a lot of this oleic acid, omega-9, but much less omega-6. So the high oleic soybean oil we generated has the highest omega-9 content and very little omega-6. I think the use of this kind of healthy oil will help to reduce cardiovascular disease risks. Soybean has been widely recognized to be really good for our health. So we generate, in addition to high oleic soybean, various other functional soybeans. And for the third problem, that the lack not enough beneficial phytonutrients, we generated tomatoes that are simultaneously enriched in GABA, in lycopene, in vitamin D, in vitamin A, lutein, alpha-carotene, vitamin C, Q10, quercetin, and folate. All of these are phytonutrients that we really need to fight inflation, to fight the oxidation, and to fight against the aging. All right. Thank you for your attention.
Thank you so much. So when I was a kid, I got truly fascinated when I learned that the genetic code is universal, that all organisms share the same genetic code. And I couldn't believe that, actually, because my father was a paleontologist and I had been taught from an early age how evolution worked and how, surely the language of genetics would change over the lengths of evolutionary time, separating bacteria, for example, from humans. But that's not the case. The genetic code is universal on the planet. And the field of, well, let's just face it, biology itself is at its heart really about information. Information is life. And I guess I have a controller here. I was told my slides didn't work. But they do. And there's the real Nobel Prize winner, this little tiny animal. So if the code is universal, if you are interested in how organisms use information, then why not study a relatively simple animal? This little animal shared roughly 3 billion years of common ancestry with humans. And in many respects, they really are, as Bob Horvitz likes to say, a tiny little human. So I'm fascinated by how organisms handle information. And there's a lot of hubris, actually, in this field. Ever since we discovered the genetic basis, the DNA code, if you will, I think there's been a lot of hubris. I think we actually know very little about how organisms really work at the informational level. And why I say that is because the history of life, and I can speak really only for myself, but I know that when I was thinking, beginning of my career, life is, the flow of information in life is mostly vertical. We typically think of inheriting information from our parents, and they inherit it from their parents, et cetera, back through time. That's actually, I think, clearly a very common misconception. Information in the biosphere flows horizontally, and that is really the key to evolution. Innovations that occur in literally trillions of parallel evolving lineages can be shared horizontally. That horizontal flow of information is key to the evolution of complexity that we see around us. And this movie illustrates this so beautifully. You're looking at an ovary. Those are eggs form. Do you see the green stuff there? Maybe we're going to need a new mic. That's mitochondria. We are a blended organism. Our cells, every eukaryotic cell, has bacterial-like entities inside of it. These are, as you can see here, the green things are membrane-bound organelles called mitochondria that contain their own DNA because their ancestor was a free-living microbe. Our cells formed by blending and taking that origin. Just think about that. Thousands of human genes are derived clearly from bacteria because we are a blended organism. All eukaryotes are. And so innovations that arise in one lineage can be captured and utilized in another lineage. So what Andrew Fire and I discovered was, quite by accident, we discovered that all living things use guided search. Why do they use guided search? Because they, like us, are living in an information, I call it an inferno of information. Information handling requires guided search. We all know that now. Now, organisms are really good at handling information. And the human body has multiple different search engines in it. Every cell in our body has a search engine. These search engines are programmable. They use a short piece of genetic code to find and regulate other genes. And in fact, right now, none of us would be sitting here if it weren't for the function of that guided search mechanism. It's literally keeping us alive because it's controlling our gene expression. So guided search is fundamental to biology. What's exciting about RNA interference, and you heard some talk of CRISPR, we can program these search engines. Now we can make medicines that are given by injection into a human, and then they flow, circulate, find the cells where they need to function. They enter into those natural search engines that already exist, they program them, and then they can precisely regulate the gene that we designed the guide RNA to target. So we can precisely control genetic information at the RNA level, and we can turn off a gene. Amazingly, these drugs are approved. There's several already. There are going to be many, many more, and they're already having a very exciting impact in the clinic. And so I'm going to stop there, but the future is very bright because these drugs are relatively inexpensive to make, long-lasting, and they can modulate gene expression for literally any type of disease as the numbers of tissues in which delivery can be addressed increases. So the future for this type of informational medicine is extremely exciting. I'll stop there. Thank you.
I will talk about inflammation, focusing on how DNA causes inflammation, which underlies many human diseases. All right, so inflammation is a phenomenon that is very well known for thousands of years. 2,000 years ago, the Roman physician Celsus defined inflammation as having redness and swelling with heat and pain. Nearly 2,000 years ago, the Chinese physician Zhang Zongjing wrote a book called Sanghanlun, which is basically the theory of injury and cold, and this forms the foundation of traditional Chinese medicine. Fast forward to today, we know a lot more of what causes inflammation in many cases. The infections by bacteria and viruses causes inflammation to our own immune system. So our immune system has two arms. One is the innate immune system, and this is the innate immunity is the first line of defense against microbial infections. Shortly after infections by, in this case, viruses, the innate immune system is quickly activated, and that rapidly controls infection. And innate immunity is also very important for instructing the other arm of the immune system, that is the adaptive immune system, that involves T cells and B cells. And the adaptive immunity is slower. It takes several days to develop, but once the adaptive immune system is activated, it's very powerful ways to eradicate infections. So then how does our immune system know if we have been infected by a virus or bacterium? And the key here turns out to be the DNA. So DNA is present in almost all microorganisms, including bacteria and viruses. And that then provides a very powerful and universal mechanism to detect microbial infections. The problem is that we also have our own DNA, which I will get to in a minute. And in fact, DNA was known to stimulate immune responses 40 years before it was known to be the genetic material. But we still didn't quite understand how DNA stimulates immune responses until quite recently. And key to this understanding is the discovery of this DNA sensor. So in 2012, my lab discovered this DNA sensor that triggers immune response. And this sensor protein turns out to be an enzyme that is activated by binding directly to double-stranded DNA. And the activated enzyme then converts GTP and ATP into a cyclic dinucleotide called cyclic GMP-AMP, or cGAMP. And this small molecule then strongly stimulates immune and inflammatory responses. So in the past decade also, work from many labs throughout the world has shown that this DNA sensing enzyme cGAS plays a central role in many physiological and pathological processes. So this enzyme is very important for host defense against microbial infections, but aberrant activation of this enzyme can lead to a variety of autoimmune and inflammatory diseases, including like lupus, long COVID, neurodegeneration, such as Parkinson's disease, ALS, Alzheimer's disease, and activation of this pathway can activate autophagy and cellular senescence, and this pathway is also very important in anti-tumor immunity. And so in immunity, the key word is really balance. So too much immune response will cause autoimmune and inflammatory disease. If you don't have enough immune response, it would cause infections and cancer. And what is fascinating about this DNA sensing enzyme cGAS is that it has to really strike a balance between in this immunity. So it turns out that cGAS can be activated by any double-stranded DNA independent of the DNA sequence. And that is very good for recognizing bacterial and viral infection, but it can also be activated by our own DNA. And the solution to this problem is that in our cells, we keep our DNA in the nucleus and in the mitochondria, and inside the nucleus, we wrap our DNA around histones that you just heard about that prevents immune response. So it's really the location of DNA that is in the cells that is very important. But under some conditions, if our own DNA gets out of mitochondria or nucleus, then it will activate cGAS and cause autoimmune diseases. And there are many, many autoimmune diseases. It's like estimated that 50 million Americans suffer from some forms of autoimmune or inflammatory diseases. So, and this becomes an even greater problem because we now live longer. And so now the average Americans live about 80 years old, but 100 years ago, the average lifespan is less than 50 years old. And because you see a big dip here in the 1920s because of 1918 flu virus, and then more recently there's a dip because of the COVID pandemic. So infectious disease, of course, is very important, but thanks to vaccines and antibiotics, we now live much longer. But then as we live longer, we have all these other diseases, cancer, autoimmune disease, heart disease, etc. And many of these are caused by so-called sterile inflammation, that is inflammation in the absence of infections. And the sterile inflammation can be caused by tissue injury, cellular stress, radiation, obesity, toxins, and another very important factor is aging. So as we age, we have many of these inflammatory conditions, and in many of these cases, it's really our own DNA that triggers inflammation. So this is my last slide, and the take-home message is very simple. Is that our own DNA can cause inflammation, and the key sensor of DNA that triggers this immune and inflammatory response is this DNA sensing enzyme, cGAS, which is activated through a very simple mechanism that is binding to DNA. This enzyme then makes this potent small molecule cGAM that strongly stimulates immune and inflammatory response. And we believe that a cGAS inhibitor can be used for treating autoimmune and inflammatory diseases, including some age-associated diseases. And this small molecule cGAM can strongly stimulate immune response, and there are efforts in trying to use this molecule or its derivative for cancer immunotherapy and also as vaccine adjuvant. Thank you very much.
It's my great honor and privilege to be speaking in front of this distinguished audience. For the last 15 years, there's a lot of interest in the use of extracellular DNA in different body fluids for diagnosis. And two areas particularly stand out. On the left is the use of this technology in pregnancy, and the second is the use of this technology for detecting cancer. So for pregnancy, it's based on a phenomenon that we first reported back in 1997, and that is during pregnancy, the fetus would release its DNA in the bloodstream of the mother. So we're able to actually convert this into a detection tool for a variety of fetal disorders, such as trisomy 21. So you can see the diagnostic accuracy for Down syndrome is over 99%. And because of this, then this technology has been rapidly translated into routine use. And to date, this platform is called NIPT, non-invasive prenatal testing, and has been used by over 100 million pregnant women worldwide. Now, in the cancer area, we started working on a cancer called nasopharyngeal cancer, or NPC for short, which is a head and neck cancer, which is very common in the southern part of China. Now, one reason why we use this as a model system is because NPC is associated with Epstein-Barr virus infection. So we imagine that we're able to use the EBV DNA as a marker for this cancer in different body fluids. And indeed, for example, you can see on the left-hand side that in individuals with this cancer, their blood plasma is stuffed full of these EBV genomes. On the right-hand side, in an individual without this cancer, then most of them are negative. So we wonder, is it possible we can actually use this for early detection of this cancer? Now, like many cancers, the earlier you detect NPC, the earlier you can do something about that. Now, for example, if you detect it in stage 1, which is the top line, then long-term survival is over 90%. But if you wait until stage 4, which is the bottom line, then long-term, some 40% of people will die. But unfortunately, in Hong Kong, where I work in, some 76% of this cancer is only discovered late. So we want to do something about that. So a few years ago, we did a large-scale trial involving over 20,000 individuals, and then we see what happens when we use the screening technology. Now, we find that if in the unscreened population, some 80% of NPC is found in a late stage. But however, in the screened population, we have shifted that to 70% is in stage 1 and stage 2. Now, because it's so early, you can see that in the top line, which is a screened population, over long term, some 97% of the individual actually survive. But however, if you don't use the screening, which is the bottom line, over long term, some 30% individual actually died. So now with the use of the screening, will greatly reduce the actually mortality from these disorders. But NPC is only our first goal. We like to be able to extend this technology to other cancer types. But of course, not every cancer is associated with a virus infection. So after testing many different approaches, we eventually arrive at the use of epigenetics as a tool. And more specifically, we start with DNA methylation. So in 2013, we actually report the use of genome-wide methylation analysis to detect cancer. And we find that by using this approach, one test is able to detect multiple types of cancer. And furthermore, because different tissues in the body have different DNA methylation profiles, I can also use this to tell us where's the cancer. So this technology has now been commercialized. We've licensed it to a company called Grail, who has launched a test called Gallery, which is the first multi-cancer early detection test. And this is undergoing trial in many parts of the world. And actually, this field is actually getting hotter now. For example, recently, Exact Science and Garden Health have also entered this area with their own products. Now, we've been thinking, how can we improve it further? And one thought is that because it's an epigenetic test, and most of the DNA sequencer, which is on the market now, are invented to look at genetic changes. But if you want to look at epigenetics, we'd have to somehow convert epigenetic change into genetic ones. And in this conversion process, it could be destructive to the DNA. It would add cost and complexity. So we're thinking, is it possible I can develop an epigenetic test without this of conversion? So we're thinking that now if you look at DNA inside cells, it's basically long chromosome length molecules. But interestingly, in the blood, it's actually fragmented into millions of pieces. And we and others have been very interested in how this fragmentation process proceeds. And this field is now called cell-free DNA fragmentomics. And there are actually many different types of fragmentomics markers. For example, you can look at the size of fragments. You can look at the end motif, the sequence there. You can see whether it is jagged or whether it's blunt end. Now, for example, in this slide here, it shows some data, which is yet to be published, about the improvement of different types of fragmentomics markers. On the x-axis are different types of fragmentation analysis, which I didn't have time to go into detail. And the y-axis is the accuracy of such tests, which is the area under the curve of the receiver operating characteristic curve. So a perfect test will have a value of 1.0. A test which is a flip of a coin will have a number of 0.5. You can see by the use of the fragmentomics approach, we can gradually improve this sort of multi-cancer early detection. Now, apart from fragmentation, recently we've all been wondering about integrity of DNA in plasma. One reason why I say that is because most individuals who sequence circulating DNA will use a short-read sequencer like this. And recently we're wondering what would happen if we use some of the newer single-molecule long-read sequencer to sequence circulating DNA. Now, for example, what will happen in the plasma of a pregnant mother? And interestingly, when we do that, you see that with short-read sequencer, most of the molecules are short, as we indicated here. But however, if you look at long-read sequencer, we can discover a previously unknown population of super long cell-free DNA. Now, the importance of this for the field would be like this. For the last 15 years, we're basically looking at short text messages sent to us by the baby and by the cancer. But now, with the discovery of super long cell-free DNA, we can see a whole Word document. So in summary, I think circulating DNA basically offers a non-invasive window into our health. And by combination of this and other newer modalities, hopefully this will act as some sort of a crystal ball into our future health. So thank you.
So 50 years ago, when I started in science, the fundamental question was to try and understand how you could pack 2 meters of DNA that we have in each of our cells into the small space of the nucleus. Obviously, understanding chromatin structure is very important because this will regulates the access to the substrate of most biological processes, DNA, and processes such as transcription, gene transcription. For the purpose of this talk, I will illustrate how we have used different structural methods to define chromatin structure and concentrate on the resolution revolution that has been recently through using cryo-EM. So the resolution revolution in EM is due to multiple developments, instrumentation and automation. We have now direct electron detectors. When we started using EM, we used to collect data on films. Now we have direct electron detectors. That means we can collect much cleaner data and much faster. And of course, computational method for image processing structure determination, such as Bayesian approaches rely on, and machine learning has vastly increased the speed at which we can determine structure. And I should point out that most of these innovations come from scientists doing basic research in academic institutions. So Karolin already showed you the structure, the nucleosome core, which organizes about 150 base pair of DNA. We started doing this work in 1976 after Roger Kornberg, who had been in the lab, had defined that the building blocks of eukaryotic nucleosome is the nucleosome. And as you can see, it took 20 years to go from the crystallization to obtaining the structure. I should mention that the method at the time for determining structure was X-ray crystallography. So the bottleneck for doing that, you need to get crystals, and you had to get ordered crystals. So it took 20 years essentially to go, or 16 years to go from 7 angstrom to atomic resolution, which was the work of Karolin and Tim Richmond. When we switched to, when I was in Singapore, we set up cryo-EM, and we thought we'll try and redetermine the structure by cryo-EM to test how quickly we could do it. Basically, we went from 20 years to in 3 months with actually a very small data set getting atomic resolution structure using cryo-EM. And it was actually at this point, up to then we have been using CPU, and it's this point, we used this data set actually to test how much we could improve the processing of the data using GPGPU like NVIDIA. And we found that switching CPU to GPGPU, we could determined structure, but 50 times faster. My own work has really concentrated on trying to understand telomere, the structure of telomeres. Telomeres are the structure that cap linear eukaryotic chromosomes, and this capping structure is very important for maintaining the integrity of chromosome, and telomeres are sort of the molecular clock for aging, and they're also involved in cancer. So the structure on the left is a structure of telomeric chromatin. I should mention that telomeres have very unique sequences, very repeated G-rich sequences. And we see this columnar structure very similar to what Karolin has observed in Archaea. And this was done by single particle EM to fairly high resolution. And when the central image is cryo-EM image of these reconstituted fibers where you see the nucleons lining up, but we see different structure. And we found actually that these structures can unstack, the nucleus can unstack to this, give this open conformation, maybe giving a clue how when you get DNA damage, and I should remind you that telomeres are a hotspot for DNA damage, and yet their structure has to be conserved, how maybe you can get DNA damage repair machinery to act on nucleosome without having to unwrap the DNA. And finally, I think that the great future for structural biology is that we now can actually determine structures of complexes inside cells. And the technology is called CLEM, coupled with tomography. CLEM stands for correlative light and EM. The idea is that you attach a fluorescence tag to your favorite, to the protein in your favorite complex, and then you image a slice of the nucleus, in this case the nucleus, in a light microscope, and you there find your complex, index it, and then go to electron microscopy. And since this is slices of cells, we do, in order to get three-dimensional information, we tilt or we do tomography of these slices, and it can be done in tissue, not just in cells. And Sarah Sandin used this approach to determine the structure of whole telomeres inside cells in various cell types. So we could see the difference of telomeric structures between different cell types. I apologize for this complex structure, but if you look at, we have a three-dimensional structure of a whole telomere. In it, you can trace chromatin fibers, and it's actually a heterogeneous structure or mesh where we see the different size of the width of chromatin fiber, and it should be 5 to 30 nanometers. So variety of structure, but very interestingly, we also see, you can see in the lower level, we see inside telomeres, inside cells, the same structure, the 13 to 14 nanometer columnar structure that we had observed doing cryo-EM of reconstituted telomeric fibers. Okay, that's all. Thank you very much.